As discussed earlier, we are actually going to talk about the formation of urine or physiology of formation of urine. But to know that, we have to get acquainted with a few details about the nephron, their locations and the blood supply. That is what we will start with in this session. So as we discussed in the earlier session, this is the Bowman's capsule, a neck followed by proximal convoluted tubule, a loop of Henle or Henle's loop, distal convoluted tubule, which then joins into collecting duct. Now that's broadly what we discussed as a nephron or a uriniferous tubule. But then these nephrons are not uniformly distributed. Some of these have major part embedded in the cortex, and just a part of this loop of Henle is extended into the pyramids. Such nephrons are called as cortical nephrons, whereas there are some in which most of this coiled part is in between the cortex and the medulla, while restored, that is majority of this Henle's loop is embedded in the medullary region or in the pyramids. These are called as the juxtamedullary nephrons. Juxtamedullary nephrons. Then these nephrons, as could be seen, they join into a common collecting duct. A number of collecting ducts, then they converge into a papillary duct, and these papillary ducts finally open into the renal pelvis through a duct of Bellini. Duct of Bellini. That is what the papillary ducts will join into, and then it will open into the renal pelvis. So as I talked about, there are two types of nephrons: cortical nephrons and juxtamedullary nephrons. Juxta actually starts for junction. It's a set of nephrons located at the junction between cortex and medulla, cortex and pyramids, let's say. So broadly, if this is the junction between cortex and medulla or cortex and pyramids, there are some nephrons. Suppose if this is the Bowman's capsule, this is the proximal convoluted tubule, this is the Henle's loop, this is distal convoluted tubule and collecting duct. This is one. Second one is Bowman's capsule, this is how it will go, join into the collecting duct. So this is the cortical nephron, wherein most of it is in the cortex. Renal tubule, Bowman's capsule, almost everything is more towards the cortex. While here you can see some part is in the cortex and a significant part of it is embedded in the pyramid, or let us say in the medulla. So this is called as juxtamedullary because it is more in the junctional region between cortex and medulla. So this is juxtamedullary nephron, juxtamedullary uriniferous tubule. That's it. Now what significance does it have? We will try to understand that. Now that is in general the nephron. Let us get to know more about the Bowman's capsule and the blood supply related to that. Okay, this is the Bowman's capsule as we have seen. Then as I've already mentioned, this is the visceral layer, this is the parietal layer. Now parietal layer is made of squamous epithelial cells, flat polygonal cells placed on a basement membrane edge to edge without any gaps in between. Whereas this visceral layer, a number of cells from it are modified into cells which are called as podocytes. Podocytes. Now, podocytes, details of this we will try and understand a little later. Then, as could be seen, this is the afferent arteriole, the incoming arteriole, which then divides into a number of capillaries. There's a cluster, there's a bunch of capillaries that gets suspended in the cavity of Bowman's capsule. This cluster of capillaries is called as glomerulus. Glomerulus. The glomerular capillaries then join and come out as an efferent arteriole. Remember, the incoming vessel is an arteriole, the outgoing vessel is also an arteriole because the oxygen content of blood has not changed significantly. So, incoming blood is arterial blood, oxygenated blood, outgoing blood is also oxygenated or arterial blood. And the two are connected by these glomerular capillaries. Capillary, it starts from the afferent vessel, goes through all this and joins directly into efferent vessel. That means there is no sub branching in between. That is why these are called as parallel capillaries. Now, between glomerulus and the visceral layer, there is a minor gap, which is called as a filtration space, filtration cavity. Next to the visceral layer, there's another cavity which we have called as Bowman's cavity or Bowman's space. This is in general the structure of Bowman's capsule and glomerulus. Together, it is called as a renal corpuscle or a Malpighian body or Malpighian corpuscle. That is glomerulus, afferent vessel, efferent vessel and Bowman's capsule almost up to neck. That is called as renal corpuscle or Malpighian body or Malpighian corpuscle. As I said, some of the visceral epithelial cells, they are modified into podocytes. This is the podocyte. As could be seen, this is the basement membrane. This layer is the basement membrane. Here is a podocyte cell with tiny finger-like projections. And what you are seeing here, it's the endothelial covering of the glomerular capillaries. Glomerular capillaries. Now, the unique feature of this endothelial covering is it has fine openings in between. These are called as the fenestrae or fenestral pores or fenestral cavities. So, endothelial lining of glomerular capillaries is said to be a fenestrated endothelial lining. Glomerular capillaries, they are lined by fenestrated endothelium, a kind of epithelium with fine pores as could be seen here. So now here is the filtration space or filtration cavity. This is the glomerular lining. Between the two is a basement membrane and of course these fine pocket-like formations here. Now what is their role? We will try and understand that when we actually try and understand formation of urine. Okay, a few more details related to the nephron or Malpighian body to be precise. Say this is the Bowman's capsule, the visceral layer. Here is the afferent vessel. This is the efferent vessel. Rest all would be capillaries connecting them. Now, here, that is this region, there will be all capillaries, and in between these capillaries there are certain cells which are known as mesangial cells. Whereas, if say this is the afferent vessel, there are certain cells in its covering, here, in this position. These are cells called as juxtaglomerular cells. Whereas, suppose this is the distal convoluted tubule, TCT. There will be some cells in this position which are now called as macula densa. Remember, all of them have unique functions to perform in the physiology of urine formation. Mesangial cells, their function is to phagocytose the particles which may be invading the process, or they could be glandular in function. Okay. Juxtaglomerular cells, they are responsible for secretion of renin, which has an important role in maintaining or regulating the blood pressure, arterial blood pressure. Macula densa would be a bunch of cells responsible for monitoring the mineral composition of the filtrate, solute composition of the filtrate. Filtrate in the sense the filtrate that's going to form the urine finally. All these together, macula densa, juxtaglomerular cells and mesangial cells, all these together, that's called as juxtaglomerular apparatus. It is all the cells together, all the machinery that we are seeing here. Called as juxtaglomerular apparatus. Now that is broadly the magnified view of renal corpuscle or Malpighian body, in which we can see different types of cells assigned different functions. Each one has an appropriate role to play in the process of urine formation. Now urine formation, when I say, I am referring to a three-step process. The first one is called as ultrafiltration, or it is also called as glomerular filtration. That's one process. Second process, it's called as selective reabsorption, and the third one is called as tubular or renal secretion. It's very interesting to note that kidneys receive around 1200 ml of blood every minute, out of which 650 ml is the plasma. 1200 ml of blood that's received by the kidneys, 650 ml of that blood is plasma, out of which 125 ml it's removed as a filtrate. Mind well, 125 ml per minute, that goes to over 180 liters in 24 hours. Whereas actually the amount of urine that's eliminated, it's around 1.2 to 1.8 liters in 24 hours. So this means more than 99% of the fluid that is collected as the initial filtrate, it's actually taken back, and hardly 1% or less would be eliminated as final urine. Now, how does that take place? We will understand that maybe in the forthcoming session. Keep watching the videos, like them, subscribe to them, share among your friends and family, and do feel free to give your suggestions or whatever you have to say about the videos. Thank you.